I request you all to keep your video on. Please put your attendance also in this chat box now. Please put your attendance in this chat box. And do not forget to put your attendance, because I will be taking this attendance when I will be marking at the end of the day. Okay, so I take this recording and from the attendance data. So please don't forget to mark it. Okay, I think we should start now. It is only 14. I think another two students will join. Anyway, I'll come to the discussion. Today, actually, my plan is to discuss how to make the research planning, what are the components there. And not only simply I'll speak not like that, I will also follow some protocol approach. The reason is that you should also know from where I am teaching, so that for some discussion you can always refer. Let me coming into our textbook. You may see, okay, I'll show you. My screen is already shared, I believe. No. I'll just share it now.
Sir.
I'm sharing now. This is the textbook. I'll show you from the very beginning. This is the research methodology Kothari. I don't like that book much, but it is at least one important thing I should appreciate is that the book is little organized. So for example, first chapter, they're talking about introduction, meaning of research, objective of research, motivation in research, type of research. These are all very important, and I will suggest you to quickly go. I will also go through them, not an issue. And then we are talking about defining the research problem. So this portion actually today will be discussing some extent on what is the research problem, how to select a problem. In our last class, we discussed how to search it, right? Search method. Of course, the entire things we cannot discuss in a day. You should be little doing some practical rather, then only you will understand how we are doing. Now, okay, so first chapter, if you go through, you will see, actually I'll be showing from the textbook, then we'll come to PPT also. So when I'm talking about defining the research problem, so you need to know what is the problems, what I know, what could be the possible problem, how should I choose that, okay? And what could be the problem in study when someone is doing something. Selecting a problem, what should you choose? If I ask you, I'll give you very simple logic. Necessity to defining the problem, technique in defining the problem. But you see, the things does not happen like that. When I am doing research now in my lab, do you think that I'll go to the book and then only I'll do? No. This idea is already coming into our mind. It is inculcated in our practice. So definitely not that we will follow these books and then we will do the research. Not necessary. Just for illustration and just for understanding very small thing, we'll be discussing about the research problem part. What is the methodology, what is the method, how the research process will go. I think I will talk about them very soon. After once I start one presentation on this, I'll come to there. So this is talking about research process. You can always stop me and you can ask about that. So today I'm going to take these presentations just for making a continuity on that, and then coming into our main discussion on how to selecting the problems. So this is of course a PPT. I don't remember some resources we downloaded. This is talking about stages of the research, how people normally do any work in a lab. So before that, you might have two sources for your information. First source may be that you are getting motivated from some topic. You have already studied lots of literature, you are gaining those informations. For example, if I ask you, can you tell me one very important problem for humankind now? Anyone and everyone, from social knowledge, from their scientific study and documentary, you all know many very important problems nowadays. One can be global warming, one can be global warming consequences. For example, increasing level of carbon dioxide. From where it is coming? Industrial activity. So people, some of the thematic area is already presented to you because you already know them, they exist. From where you are knowing? You know from research, someone else research, you know from your communication channels, maybe televisions, maybe news, even social media. You are already aware the impeding problems, the very bigger picture. Once you have the bigger picture, people normally look into smaller picture. So it is not that you will reach the sustainable goal, attaining the sustainable goal, right? People are targeting, for example, India is also targeting some 2050, I believe. I was checking yesterday's news. So that means we are still having some 30 years time period to make it zero emission binding. All countries are promising like that. Whenever there is an intergovernment panel in climate change, IPCC meetings are happening on climate change, they will promise. Of course, this promise has impact. So any government organization will also make some protocol prioritizing some area, prioritization. So when you will get some, for example, we are having in India, we are having the government bodies like Department of Biotechnology, Department of Science and Technology. They also approve some research proposals which should be commensurate with our government policy and government goal. So research is somehow defined. It is not completely undefined. It is having some use. So if I ask you simply, can you propose a research domain? So basically I'm talking about the domain. Domain is already known. You will try to do some research on, maybe on healthcare, maybe on energy, maybe on environment, reducing carbon, environment, maybe producing more biogas, energy production, more biogas production, or bioenergy production, biofuel production. The area is well known. What is not known is that, have you identified the questions? That is very important. So domain is known, but the question is not known. So mostly we do the problems, we do not do, most of us, we do not do from problem solving. Solving any and every problem is not the approach. What does it mean? So do you think, for example, when you are coming from the day one, you are waking up from your bed, and at the end of the day, you are also going back to bed. So you are working, you are in the lab, you are coming to research office, or you are doing some activities, you are going some organization. But at the end, you must be facing many difficulties in your day. Everything is not smooth. So do you think that all the problems you are identifying, you are doing on research on them? No, many of you are not doing that way. The first reason is that possibly you are not identifying the problem itself. And second one, possibly you do not agree that they need to be addressed. So first of all, when you are doing anyone proposing anything, they need to understand the questions. And questions should be very small, very specific. You cannot do that global warming you are going to change by a research work. No, you cannot. You can only contribute a very small fragment to attain that. So that means you must have very clear questions in your mind. Whether are you going to solve something? For example, you are going to prepare some nanosorbent which can actually degrade some carcinogenic dye. So maybe in industries, just a second please. Pema, can you close this door? The outer one, not this one also. Okay. So basically we are talking about the situations, and you need to identify your problem very clearly. So for example, you are looking for solving some azo dye. In azo dye, where is it used? Very widely used in textile industries. And possibly you also know there is a lot of problem there. Now question comes, not only azodite, there are many heavy metals are also there. You know what is the important heavy metal found in these kind of industrial effluents containing azodite, textile industries? One important is chromium, hexavalent chromium. That we know because we are studying like that. But basically, you will also find these problems in many magazine, in news channels. These problems will come. Now, do you think that you are going to solve the entire problem? No. So for any research, you should have very small sets of questions. So now I'm taking today, for example, showing you, I'm taking a question, how can I remove, how can I treat the azodite which is coming into wastewater? So my question is very clear. I want to remove azodite, for example, wastewater. Now immediately many options will come into your mind. Okay, what can I do? First option you will say, sir, can I replace the azodite with some other dyes which is more environment friendly? Biodegradable dye can you use? Can you use some biodegradable material which will be stable also? Because you need to talk about stability. Because when I am using some fabric, some dye, they should be stable, right? One time wash, if they're going away, then customer will not be there. So essentially, that is one approach. That is, for example, can you do for some green synthesis method? Greener approach for production of the dyes, that is more appreciable. But for example, you cannot have that control. You have to only solve the recent problem. So maybe I'll look for some solution. What could be the solution? So maybe can you degrade it? So basically, when I'm asking the question itself, there is a lots of studies being done. How can I address a problem? Can I degrade it? You say, no, sir, this is not biodegradable. This is very toxic chemicals. The molecular structure is very hard to break down by microorganism. Azo compound is there. Nitrogenous sulfur is there. Possibly it cannot be broken. Maybe. I'm not talking about that it cannot be broken, but assuming. So what is the alternate option? Can you photodegrade it, chemically degrade it? Can you use Fenton reagent? Can you oxidizing agent? So many questions comes and you have to shortlist. No, I cannot do. Last option, can you adsorb it by using something so that I can remove it and I can replace? So for example, yes, possibly you are thinking that I want to use some adsorbent. Then you should ask what kind of adsorbent I will use. So you need to think why your material will adsorb that. So you are thinking about the molecular structure, maybe azo, so amino group will be there. Amino group means positively charged. So I should look for something material which is negatively charged so that they will attract. That means these are all hypotheses. You have not done anything till now. So that means hypothesis is very important when you are going to get these things to be done. So question identified. So for example, you identified the questions that I want to have some nanomaterial, not some. Then there can be n number of material there. So you are already shortlisting. No, I will only use the nanomaterial which is easily available, low cost, not environmentally dangerous, which is known to be a little bit effective also. So I'll have some 10 member, but I need to screen them possibly. So you have something already in mind that I want to do that. I will use nanomaterial and I want to see whether it can remove it or not. Now there will be some hypothesis. This is very, very important for any researcher, how we are preparing hypothesis. You may find that I'm not moving much in the slides, right? Anyway, that is not an issue. I can move. Hypothesis is actually your assertion, your assumption, presumption that okay, this I do, then that will happen. That means I'm doing some presumptive approach on mechanistic behavior or outcome. That means I am presuming something. So when I am doing, for example, I am choosing some nanomaterial for removal of some azodite. What is my presumption? My presumption is that I can produce some nanomaterial. I'm also assuming that if there is a water containing the azodite, if I put the nanomaterial, they will come and bind. These are all your hypotheses. Not necessarily they will do like that. And then I can take out this nanomaterial and separate it out. So you may say, sir, this is obvious, na? Yeah, it is obvious, but you cannot guarantee that it will work. Your nanomaterial may actually become unstable in the solution. It can break down. The azo dye may not bind at all, or maybe they need some other pH. So you are already thinking process is going on. So hypothesis means you are hypothesizing few things. I am hypothesizing that my nanomaterial would be stable in that pH, it will not dissolve, and the azo dye will come and bind. So we can clean the water from the azo dye. If I put the nanomaterial, they will absorb, and then I will separate them by centrifugation or filtration. That means I already see this is called hypothesis. I already see this is likely to happen. But how can I prove that? I have to prove also. So proving the hypothesis is the next part. And to prove it, you need to do something. Some steps you need to have, strong steps. That is called methodology. What approach you will take? Let me tell you one very simple example. I'll take one slide again, then I'll come. I think you understand that we are moving. So from question to hypothesis, hypothesis means working principle we are proposing. Then I will have research plan. Research plan is how I am going to do that, planning. So if I ask you, and I assume that this class is not my class and you are all present here, so can you respond that how can I validate that? You may say, sir, you first prepare the nanoparticle in lab using some protocol. Okay, I prepared. Bring some water from wastewater containing this dye. Okay, I'm bringing. Then what you will do? Then you will say, sir, I will take the water, I'll put the nanoparticle, and then I may find that water is becoming very clean and I'm separating the nanoparticle, right? And I'm finding water is very clean. Very clean means, what do you mean by very clean? You will certify or you will vouch like that? You will take an oath on very clean? No, you have to measure, right? So you are planning that, okay, the azo dye is absorbing some wavelength, so I will measure its absorbance, for example, at 650 nanometer or like that. So before I am adding any adsorbent, I'll also have another vessel, water. I'm not putting any adsorbent, and I'll also measure. And for example, if I find this absorbance is low, for example, I'm getting only 0.3 and here absorbance is 0.8, that means the dye has been adsorbed. So what is methodology? Methodology is a plan you are going to have to validate or invalidate your hypothesis. Does not guarantee that what you thought it will happen. But for example, I'm doing this. I'm taking this one and I'm putting nanoparticle, and I think my dye absorbance is coming down. My first question, please listen very carefully. I'm having some wastewater, I'm putting some nanomaterial. After 1 hour, I find that the absorbance of the liquid is coming down, absorbance is falling. Can I say that it is being absorbed by the nanomaterial? Can you tell it, confirm? I think you are listening my questions, right? Can I get any response from you?
Yes.
Response, please. No idea on that?
Yes, sir.
No, you see, what is my hypothesis? My hypothesis is that nanoparticle will adsorb the dye. How can you validate? You can only validate if you do something, na? For example, if I'm saying that I'm having one, you are having one vehicle and your friend is having one vehicle, and your friend is telling you, no, my vehicle is gives very good mileage. How can you prove that? First, I'll bring both the fellows together. I will clean their tanks. There should be no fuel. Then I will give everyone 1 liter, 1 liter. Then I'll ask them to go for some time and show me how much mileage they can show, right? How much kilometer they're going. So you have to prove it. And for example, at the end, you see both are coming same value. What does it mean? What you hypothesize that your vehicle is good or better or best, nothing is valid. So similarly, every experiment need to be validated, any hypothesis. So hypothesis, how can I validate? I will take water, I will put nanoparticle. After 1 hour, I'm seeing color has gone down. Color has come down. I can say, it has absorbed. Yes or no?
Yes, sir.
Yeah. Normally, I will say like that. But if I am very much enemy of you, for example, this work comes to me for review, and I say no, how can you guarantee that? I may assume that okay, dye is getting removed, but I may say that dye is getting photo oxidation because we are doing the work in lab, light is there, and your dye is getting degraded by light, not by absorption. I may say, na. Then how can you validate that? You got my point? There should be, your research design should have all possible counter defense. You should always be ready. I may ask you, no, nanoparticle is not absorbing. Your dye is simply getting degraded automatically by light. So then you say, yeah, you cannot 100% guarantee that nanoparticle has removed, not light. How can you justify your point? Then you will say, sir, you look, sir, this is one set I am taking. I will take another set also. I will add nothing. I will not add any nanoparticle. After 1 hour, you see, sir, the color is not changing. Then I will appreciate, yes. It is not photocatalytic. It is only being absorbed. You are getting my point? So I need to have some study, some experimental set, some design to prove and to disprove all the hypotheses you are going to make. Am I clear? It is your responsibility to validate whether really absorption is happening or not. So what I will do, I'll take one batch, I will add nanoparticle, I will take also another batch, I will add nothing. I will not add any nanoparticle, and I will show you, you see, after adding nanoparticle, something is happening. So what is normal? You see, when I am adding nanoparticle and I see the decrease in color, that is called cause and effect. This is a very important model for any research. Cause and effect. If you do not do something, then you do not expect something change also. Everywhere, right? Everywhere there is a cause and effect. If you take your lunch time in lunch, then only your hunger will go. So cause is there, I'm putting the food, hunger is gone. Maybe you are measuring blood sugar. After taking food, my blood sugar increases. So there is always a cause and there is also one effect. Okay, that is called cause-effect relationship. But there may be some relationship that is merely casual, nothing to do. Some relationship may be merely casual. What does it mean? As I told you, putting nanoparticle, dye color is changing, but it may be simply dye is not being taken by nanoparticle, but maybe simply their photo oxidation is happening. So if I claim that addition of nanoparticle is causing this dye color change, and if it is not really happening, actually photo degradation is happening, then that relationship known as casual relationship. Any research work, I need to know this cause effect and casual. Casual means not really important, it's simply happening. Okay, it's simply happening. Mostly all, for example, all holidays is coming on Thursday and Friday, or maybe on Sunday. If it happens, do you want to blame the government for that? That no, all puja, Deepavali, everything is coming, for example, on Sunday. You see, this is not connected to anything. This is actually casual. Maybe I'm observing that this year all holidays in coming on Sunday. If there is no background information, there is no manipulations, and still it is happening, then I will say it is a casual relationship. Casual means no reason, simply happening. I cannot attribute any logic behind that. That is called casual. So casual relationship must be avoided. cause effect model must be evaluated when I'm doing a research. So I must have a research plan. What is my plan in this case? I told you I will take 2 different batch of flask. Then I'm planning to add nanoparticle in one case. Another case I will not add any nanoparticle. Then I will wait for 1 hour. I'll also switch on the lights to see that light is not the cause for degradation. So I need to have a proper planning. Research plans should be there. And then I have to collect the data. Data collection means what you will collect? You will collect how the color of the dye is changing with time. So every 5 minutes, every 20 minutes, you are measuring the absorbance of the solution. That is your data. And then you have to analyze. Then you have to see side by side your data graph for with nanoparticle, without nanoparticle. You have to see that data. Once you see this data, once the result has been done, analyzed, I will analyze this result, for example, then I will make a conclusion. Conclusion means final comment. Maybe I may make a final comment. Yes, nanoparticle of these nanoparticle can actually absorb the dye. Or I may say, no, nanoparticle is not very important here. Actually, photocatalytic degradation is taking place. So whatever may be the true, I don't know earlier, but I am having some hypothesis and I am going to follow. So these are the all stages of scientific method. Do you think that you have to memorize it now? I will say yes, because you are going to write an exam also, unfortunately. So some of the steps, for example, if I'm doing, we'll go to have the questions first, then I will make a hypothesis. Hypothesis means working model. It is not validated, but you are planning, it will absorb, so my dye color will go out. That is your hypothesis. You have not seen, but your intuition, your background knowledge, your approach is going to tell you this is likely to happen. Likely. To test it, you need to plan. All research are planned to support the hypothesis or cancel the hypothesis, right? Either I will approve it, yes, my hypothesis is correct, or no, your hypothesis is wrong. Anyway, you can do the job. So that is, to solve this, we are making a research plan. Okay, how I am taking, when I will collect the data, how I will collect the data, how much liter water I will take, how much milligram nanoparticle I will take. Everything should be known, right? When you are coming to lab, you cannot say, sir, I'll put how much, you will ask me. Na? Before you ask me, you have to note down that, okay, I need to have this much. Analysis of result, you need to know the result, how to take the result, which instrument I should use, how much solution I should use. All those things should be in your research plan. Once the data is done, data is collected, then you can sit and you can analyze the result. From the analysis, you will make some conclusions, which is more appropriate for your study. Now, okay, here I'll be talking about steps in conducting research. The specific steps guide the research process. Steps means I already told you the steps. Some people will say some 12 steps of research. There are many variants are there in textbook, but it not necessarily that you have to follow some strict guidelines. Number of steps is indeterminate, so we cannot really tell how many steps are there. Various steps may be combined, so maybe you are doing 2 or 3 analysis, you can combine together. Order of steps may vary somewhat. So for example, in my study, I am measuring the dye before adding the nanoparticle. You may only measure the dye after adding of nanoparticle. You may measure the absorbance after your whole study is over, or you may measure absorbance after every sample you are taking. So there is no strictness on how you are going to do. And importance of specific steps is variable. So we need to know whether this is really important in our study, but in maybe in another study, it cannot be very important. It may just small work. So for example, when I'm doing the dye study, then absorbance measurement is very, very important, because this is the only thing I'm doing. But when I'm doing a very big picture of wastewater treatment and I'm only measuring the dye just to know how much has been removed, and it is not very important area for me, then it will become less priority. So we need to understand there can be multiple steps and steps involved in research. In this slide, we are talking about the 12 step of research. Please listen. And this is not a theory class, that's why I'm trying to repeatedly tell. These 12 steps, not necessarily that these are the golden standard, not like that. People normally argue and represented one type, not very strictly. If you do not follow the 12 steps, or if you ask me, sir, you are doing research for last 15 years, 20 years, do you remember all the 12 steps? My simple answer is no, I don't remember. Okay, I don't remember. I even don't know all the 12 steps. When I'm seeing the slide, I will possibly recognize yes, yes, yes. So what I mean to say is that 12 steps may not be really important, but when you do a research, you will appreciate, yes, we are also following those steps, either knowingly or unknowingly. Maybe I'm not giving the name, but I'm doing. What is that? First one, identifying the research question that everyone will do. You are going to isolate some bioactive compounds for some plants. Why should you do that? What kind of bioactive compound you should look for? Do you think that we should look for some antacid, or you look for some maybe painkiller from plant source now? I will say yes, one way true, but at the same time I will say no also, for another reason. What is another reason? We are having plentiful of drugs, and these drugs is being more than 50 years, 60 years. There are many kinds of non-steroidal drugs are there, which we are using with not much difficulty, not much toxicity. Okay, we are using it. Of course, you may say, sir, plant-based material will be good. Yeah, of course, I appreciate. But we should look for those things where drugs are not available much. Okay, where drugs are not available much. So you need to prioritize your research, not simply doing another one time. Prioritize. What could be the prioritization? You should look for where there is something not there. I may say, okay, for example, do you think that we are having antiviral drugs effective in our drug regime? No, we do not really have large amount of antiviral drugs. We may look for that. Anti-cancer treatment, yeah, anti-cancer drugs are there, but not very effective till date. So variable efficiency. Why don't you look for some drug which can have anti-cancer property for some specific cancer type? So that means we are already telling you the prioritized area where medication may be look important. If you simply identify some antioxidant, all those property, okay, that is not going to be very important for anyone. So look for the questions, what you are looking for, research questions. Why I am looking for plant-based material? Simply, if I ask you, you are telling, sir, I am going to identify some drug which can work against lung cancer. I'm looking for these plants. First question I'll say, why? Did you get this on your dream? This is very important question. Don't daydream about research. So if you are talking that, okay, I'm going to use this plant for lung cancer treatment, you should have some logic. Some distance logic is also fine. What logic? Now you may say, sir, this drug has been known to reduce the cell growth. This drug someone has studied, not you have not studied, someone has studied that this plant extract have potential on anti-inflammatory response. Someone might have studied that it can having the possibility of reducing the growth of animal cell lines, particularly lung cell lines. That means some data is already there which is indicative, and that is very, very important. That is going to tell you initial review of literature. You need to have something. Okay, something should be available so that you can justify, yes, this plant may be useful. Then you have to distill the questions of research problem. So that means there are so many questions. You cannot do everything in your lab in your lifetime. You have to find out what you are exactly going to do. So do you think that you are going to do all 1,000, 10,000 plants you are going to analyze? No. You may say, no, sir, it is not possible for me. I will take only 5 plants, for example. And what study you will do? So cellular study, you are growing some lung cancer cell line, and then you are putting this, and then I want to see the cancer growth is happening or not. That is only your things you are going to do. You say, if you say yes, okay, that means your question is now fixed. And then you will go further. How the people have already done on this, more detailed. This will be more detailed. How people have studied, what kind of cell line they have studied, how long they have studied, how they added the drug. So we will go for literature to know everything. What is the method? How should you do it? Some idea we should get from literature. Then you formulate your hypothesis. Hypothesis means you will say, sir, I will grow one plate of tissue, lung cancer cell line, I will add nothing. I will add another cell line where I will add my plant extract, and another line maybe I'm having where I'm giving some standard drug which is useful, known to be useful. So that means you are already planning, you are formulating the hypothesis. Then you are determining the basic research approach. Approach means how we are going to do. I told you how you should follow those works. What is the basic plan? How we are going to analyze the cell? How can you measure the cell line? You are saying cell may not grow. How will you measure the growth? Every question should have a how you must respond to each of these queries. Why you must respond. Why I am adding? Why should I grow for 24 hours or 12 hours? Why not 36 hours? All questions you should be having response. So any design, any plan you are doing, it must have a logic, and that should be the basic research approach should be telling you. Identifying the population and sample. That means on which things you are studying. My population is cell line. What cell line? For example, I'm going to say human cell line. This is your population. You may say, no, I'll also study on guinea pig. I'll also study on rat. That means these are all things on which you are going to test. So rat, guinea pig, human cell line, or maybe bacterial cell, whatever you are going to do. These are all your population. You are going to validate your study on them. And sample. Sample means how long you will do and how many times you will take sample. Sample for what? For cell. How many cells are growing? So cellular measurement. You may go for cell measurement. You may go for cell activity measurement. So how much frequently I should take a sample? You should have a plan. That is going to lead you for designing the data collection, how you are going to do that. It will tell you every 2 hours, every 4 hours you are going to do. Then you are having the data collection instrument. If it is instrumental method, how I'll give measuring the cell amount? So you may say, sir, I can measure in under microscopy, or I will use some fax method, okay, cell sorting approach. So you need to know, you need to understand how I'm going to do. Then you have to do the implementing the research plan. Till now I was talking in pen and paper, and now you have to live and you have to go to the lab, do the job. Once all these things are followed, you can come and do the interpretation of result. Now, if I ask you, after reading these all 12 points, do you think anything has changed in my mind? No, you already know them. Only difference is that I do not specifically point-wise, I did not know all the 12 steps we are going to use. Okay, so these things are really important. First of them is the research questions. All it begins with the questions. How, why, when, where. You should have the questions. Any study you are going to do, it will have a question. Definitely not necessarily that you will put a question marks, because question is already there. Possibly you are looking for the right questions and right answer. And from where the question will comes? From curiosity. You are interested. Every people are different. So you might have some more interest on something. Information gap. You find something is missing. Some controversial topic, maybe, or global problem topic, maybe. You are reading it from literature review something, and it came into your mind that people are not doing like this. Yeah, that is the question you should always address. You should always look for into it. And other people, maybe some motivations. You may be getting motivated by some worker, some Nobel laureate study, your guide study, your friends' work, how my juniors are doing or my colleagues are doing. We can also find out some problem they might not have seen, but you may see. I think time is not sufficient enough to continue all the things, but basically I'll stop it here, where I'll be talking about the descriptive questions. Questions should be written. Relationship questions, that means I'm going to talk about them, and difference, how they are going to be unique from one another. So these questions could be always asked whenever I'm going to choose any data analysis plan. Particularly if I take example, then only it will be more easy for you to understand these queries. Descriptive questions asking for what is, asking questions on the research participant, testing, measuring their performance, survey research. So we can also have something descriptive approach. Do you think that all research is done on lab? No, there are certain kind of research is also done. I will talk about them more detail. These are known as survey. Some studies are known as horizontal study. Okay, some studies are known as cohort studies. I'll talk about them. Research design. Some study is only done by getting information from people's behavior. So for example, I want to know, is Indian people are following the right etiquette of hand hygiene in this corona period? If I want to know that, is that question very wrong? No, question is right. Why? Because following right hand etiquette is really important. How can you do that? So do you think that you will go into some busy shopping place and you will observe how many people are doing like that? Yeah, you can do that. That is also known as observational research. But you cannot poke them. No, you should hand sanitize. Then that means you are actually intervening. So research design can have many approaches. You can do some intervention. So you may say, okay, if some scholar is going and telling the people, is there any change in their behavior? That can also be your research study. So basically, there is no limit what kind of study you can do. But if you are going to have one question, you should follow it. If I want to see only how people are behaving after this first or second wave of corona, how people are maintaining hand hygiene, you should make many approach. One approach is that you are having many people recruiting, you are sending across the city and you want to observe. So they will observe, note down. I'm counting 500 people I've seen. Out of that, 300 people are actually putting their mask, 200 is rubbing their hand. Some data, observational data. Or you can give these questionnaires in social media. Okay, you can put these questions into your friends who can respond you. You can ask, are you using your sanitizer regularly now? They may respond yes or no. Some may not give response at all. So research cannot be always in a lab. Some research can be done outside the lab, which can be based on statistics, based on existing data, based on feedback. Observational study can be more. Some study are observational study. Some study are interventional study. So if I say use of hand sanitizer is good, if I ask you simple questions, how can you prove that? If I simply say, how can you prove? Then I have to tell some 50 fellow that, okay, you use hand sanitizer regularly. I have to recruit another 50 fellow. I have to tell, do not use hand sanitizer. So do you think that kind of research should I do? No, because there is also some research ethics. I cannot say some people during this pandemic that I want to validate that using hand sanitizer corona infection rate is low. So I am having 100 students, I will tell them use hand sanitizer. Another 100 I will tell, hey, you roam like anything, don't use anything, hand sanitizer or mask. Can I do that? No. So even if there is a possibility of doing some study, possibility of doing some intervention means you are designing. So I'm telling some people to do, some telling not to do. I can decide. But all study cannot be done. Some studies are actually ethically wrong, so you cannot do their interventional study. You can only observational study you can do. Observational means you are silent. You are simply watching, okay, these fellows are not wearing gloves. Then after 1 month, you are noting, oh, he got corona. So you can note down the cause-effect relation and you can do this job. Anyway, I'll talk about those research design not today. If I speak, then we cannot have time for that. So I'll stop it here right now. I will ask you to go through the slides also, and what we are discussing in this class, please pay attention. Any questions you have quickly, you can ask. Otherwise, I will leave this.
No, sir.